Good afternoon, and thank you very much for attending this session. The first you will see this slide during this afternoon a few times, and I'm going to start on a basic level with the genes, and I would like to give you some ideas on how you could use genetics data in social science, and what the methods are and what challenges these methods pose, and also show some other related methods which can be used. The story starts in 1953 when these gentlemen, Ross Wilkins, discovered the structure of the DNA molecule, and I'm just saying that this is a famous double helix, which is a phosphate sugar backbone with 4 bases attached to them, which make pairs according to a known rule. So this DNA molecule is what we are concerned about at the moment, and just briefly about its role in the cell. It takes part in the storage and the multiplication of the information, but what we are interested in during this presentation is the expression of the information. It happens through a messenger molecule. DNA never leaves the nucleus. This messenger goes to the cytoplasm, and there will be a protein translated from this which fulfills its function. Now, how does it happen? So how does it happen? This information translation happens through the genetic code, and I think you are quite familiar with this. So I just would like to say that the machinery which is doing the translation reads these DNA chains in 3 groups, 3-letter groups, and they are called codons. And each codon, each 3-letter, can be found in the genetic code and corresponds to one of the 20 amino acids, or a stop or a start codon. So you can take any chain, and it's good fun to try to translate them into an amino acid chain which is called the protein. And it goes on until this machinery reaches the stop, and that's the end of the chain. So individual gene sequences have been published after the discovery of the DNA for many, many years, but it was in a need. There was a need for putting this into a context, into the whole genome's context, and that's when the Human Genome Project started in 1990 to sequence the whole human DNA. It happened. It finished by 2003, and then since then several other genomes, individual genomes, have been sequenced, and it turned out that this is the number of 3.3 billion base pairs we humans have, and they constitute roughly 25,000 genes. You think about the sequence between the start and the stop codon, but these genes only make up less than 5% of the genome. So most of the DNA chain's function wasn't really well known, and also they are quite identical. More than 99.9% of any 2 humans' DNA is the same. So if we are looking for the variance between humans, then we have to go for that 0.1%. So I wrote down, mentioned a few possible variations between individuals according to their sizes, and I just mentioned the biggest one, which are cross rearrangements within chromosomes or large chunks in the DNA, and they can be observed by microscope, and also they quite often mean very, very serious genetic diseases. On the other end of the scale, there is the single nucleotide change, when we only replaced one base, one nucleotide, with another one. They are very, very common, to the extent that they have their own databases, where they listed according to RS numbers, and they called single nucleotide polymorphisms, and we pronounce them as SNP. And also, their frequency is also listed in those databases because they are not present with the same frequency in populations. Now, mentioning frequency, there is a distinction between mutation, which is less than 1% in a population, or polymorphism, which is equal or greater than 1%, but this is just terminology. Now, what do these single nucleotide changes do? And I'm going to focus onto them only for the rest of the presentation. If we go back to our original DNA chain, but what it was translated into a protein, we can see that sometimes when I replace this initial C with a G, according to the genetic code, the coded amino acid doesn't change. So this is the same sense mutation. On the other hand, so it's very unlikely to disturb the function of the protein. On the other hand, if I introduce a C instead of an A here, that the amino acid threonine will change to proline, which are two very different, chemically very different amino acids. So depending on where this changes, the function of the protein can be disturbed. But I can do something even more rude, and instead of an amino acid, I make a change which will introduce a stop codon. There will be no amino acid translated from this, so it definitely will disturb the function of the protein. Or what I can do is insert an extra base here, which will disturb all the codons behind it and misplace the stop codon somewhere. Now, depending again, this is a completely different protein, might not be able to fulfill the original function at all. Unfortunately, as I mentioned, this is just 5% of the genome, so these SNPs can occur anywhere in the genome, and not all the genomic sequences are described well enough for us to be able to figure out what that SNP does. Obviously, if it's in somewhere in the regulatory regions of genes, so close proximity to the genes, then we can somehow infer. But there are large intergenic regions with unknown function, and they occur there as well, and from there it's very difficult to say what they do. Now, thinking about what I just said about their possibility to disturb the function of the protein they are located in, we can use these SNPs on genetic association studies, which we see correlations between these genetic variants and trait differences now on a population scale. For this, I need to introduce two definitions: phenotype and allele. Phenotype is, I'm sure you're all very familiar with it, any observable characteristic you are interested in. I used an example as cholesterol level measured from the blood, but can be anything. We'll discuss this in a moment. Now, the other important determination is the allele. And in this context, I'm going to use it to denote one of the several variants of the SNP. Now, if I take this example from the database RS130064, it appears to have in two varieties, two alleles A and G. This causes a glutaminylation change in a certain gene. This, unfortunately, is quite an infrequent one, because only 0.5% of the population they have measured had it, whereas the G is quite common. But it gives me the opportunity to say that the minor allele, the least frequent, is called minor allele, whereas the other one is denoted as major allele. Now, because we are diploid creatures, when we talk about these SNPs and variants, we always have to think that we have two copies of the entire set of the genome. Therefore, we have two copies of those SNPs. So it can happen that individual one has two GGs in this particular position of the genome, and because this GG is very frequent, we have to go down to look for several individuals until we probably will find one with an AG or an AA genotype. So the particular collection of this variant is the genotype of an individual. Now, this is much more familiar to you from now on. I just had this little example to show you how you can use this genetics data for a genetic association study. So in this, the measured phenotype was the high density lipoprotein cholesterol level in the blood, and I chose one SNP from the database. But this choosing this SNP wasn't random, because candidate association gene study is always hypothesis driven. I had a reason to choose this particular one. This one is less imbalanced, the frequency between the minor and the major allele, 20 versus 80%. And why I chose this one is because it is in a gene, regulatory region, which takes part in a cholesterol transport. So, the hypothesis here, because this is in a regulatory region, it might disturb somehow the gene's expression. Therefore, it can be measured in the blood cholesterol level. And indeed, this appears to be the case, because if I regress the minor allele dosage against the cholesterol level, it appears that people having more dosage of the minor allele is associated with decreased level of cholesterol. Now, this was just a simple linear regression, but you can use any statistical method. It depends on the program, or covariates if you like. There wasn't any in this one, but again, you choose covariates based on the literature. Like older people tend to have higher cholesterol, or women, or whatever. So this is very neat, but and it's quite a highly significant result. You can see on top, but variance, phenotypic variance explained is quite small. So on its own, it's probably not enough. And it would be wouldn't be very wise to suppose that any gene acts on its own. So nothing prevents us to use more than one SNP at a time. So this is where I introduce the genome-wide association studies, where I can use more than one, up to 4 million SNPs at a time, and you can buy platforms for that. The only thing I have to mention here is a new definition, which is linkage disequilibrium. It just shows that these SNPs are quite unevenly distributed in the human genome. Some of them are quite close to each other to the extent that they tend to be inherited together. Therefore, I only need to choose a few SNPs from that region to be able to infer the rest. Therefore, I don't need to genotype every single SNP to have a very good coverage of the genome. Now, these platforms are, as I say, available, and depends on whether you want a good coverage of the genome, or you want to include some SNPs which have been associated with certain disorders. It's up to you. It's quite pricey, and I don't remember, is it £200 now, 50 or something for an individual to be genotyped, so it's not cheap, but available. So you have someone for these chips had made the genotyping work, and you have access to the GWAS data. Now, this is before we choose a phenotype and run any analysis. It's always a good idea to have a quick check whether the genetics data have been QC'd properly. It's usually not an issue, because the data provider will do it for you, but because it's a chemical reaction, it can go wrong. They also can do the QC in individuals, because there will be individuals in a population with chromosome abnormalities, or who are related to each other, which is not suitable for GWAS, because we assume unrelated individuals. Now, what you can do is check for population stratification, because there might be substructures within populations. It's probably not that prevalent here in England, but if you use international data, it might be useful, and it's very easy to correct for that. Now, the phenotype is much more of your area of interest. I just mentioned that it's worth checking whether it is behaving statistically the way it is expected, like check the distribution variance or whatever you can think of. And I also wrote down the IDs of behaving statistically appropriately. What I mean by this is that because these GWAS things are quite expensive, at first glance it might be a good idea to select and invite those individuals who actually, if it's a longitudinal study, took part in several ways, but that is not going to be a good representation of the population. Now, let's suppose I've checked everything, and I've run a GWAS study. It's again the same, just the number of the SNPs change. Now, in this graph, you can see GWAS result of an entire GWAS study in one graph. This is the negative log p-value of all the SNPs, and these are the number of chromosomes, and I've got 2.5 million dots here, but you can't see them, but that's how it's meant to be. And before I do anything, this helps me to have a quick look whether there is something interesting, something exceeding the genome-wide significant level. Before going into details, I'm just saying that I only checked 2 of the top. It's the most significant result, and something on chromosome 15 also very significant to see. And they make sense because both of them are within regulatory regions of genes taking part in cholesterol transport. So at first glance, it looks good. Now, how do I interpret all of this? Now, I mentioned this genome-wide significant result. You always have to keep it in mind that you are dealing with a large number of SNPs, large number of independent tests. Therefore, it's very important to set a threshold which we accept as genome-wide significant. And usually it's 5 times 10 to minus 8, which someone calculated. Anything beyond that is not saying that this is certainly true association, but worth noticing an investigation. Those 2 dots on the graph highlighted, yes, they exceed that, so fantastic. Now, the next step is when the hypothesis-free approach is going to be paid for, because it is very difficult to interpret the results. You have to check databases, bioinformatics, whatever tools you can find to justify whether these are real, or try to assess whether these are real findings or just false positives. And as I just said, these genes, these SNPs are associated with, looks fine because they all take part in the cholesterol transport. Can I find similar studies in the literature? There are databases for GWAS studies, and both of these SNPs came up highly significantly in many studies associated with various biomarkers. So it's always, it's again very encouraging. Then I can check what is the role of the allele. This is not going to change an amino acid because it's a regulatory region, but does it have an effect on transcription or anything of the gene? It's worth checking as well. So based on all of this, the chromosome 15 and 16 results are fine. But if I go back, there is something on chromosome 25, which is the homologous region between X and Y, on its own in the top right corner. I didn't find anything about, so I can't comment on that. Now, and this was a very neat phenotype. Also, sometimes when you've got a little bit fuzzier phenotype, then worth considering to go a little bit up and 10 to minus 5 p-value to check on the suggestive level. So on this region to see if there's anything interesting. Be a little bit less strict. And also we have to keep it in my mind that if I look up any SNP in a database and I don't find anything saying that I don't know what the function, they just say that there's no function associated with it, it still can be indication of another LD, an indirect association as well. Now, the last thing I want to mention is that very important to notice that if a SNP disturbs the protein function, it doesn't mean that we can observe it in the phenotype. It depends on the phenotype what we are choosing. With the cholesterol level, a simple molecule carrying the cholesterol at various places in the blood, yes, it was observable. But it depends how many pathways are involved in making up that phenotype. If it is just a few, then yes, we will have very nice and neat results. But if it is several pathways, then it will become a little bit blurrier. And what I know about social science phenotypes, they tend to be very, very complex ones, which means that whatever phenotype they are investigating is very likely to be affected by several genes with very small effects, and they interact with each other and also with the environment. So this is very, very difficult to measure, but not impossible. Here is an example. The major depression is quite a huge consortium investigating it, and twin studies indicated that its phenotypic variance, 37% of the phenotypic variance can be explained by genetic factors. Now they conducted a huge mega GWAS for nearly 10,000 cases, and they didn't find a single GWAS level significantly. So that's how it looks in practice. Therefore, what you can do is to take very, very good care of the phenotype, because you can improve the GWAS study by well, I was lamenting on how well divided and how incident it is in a population, but I think the most important thing is to suspect the phenotype, what you are about to measure, has a biological component. You can somehow relate it back to pathways. The genotype of the data is also important, although, as I said, there are very good quality data out, so you don't need to worry about. The only thing to consider here is that, as I said, they are not equally frequent. So it can be, if you include very infrequent alleles in the analysis, then it can give strange results. As I just said, we always have to correct for multiple testing to see where we are with the data. But this is where normally this depression or any social phenotype results diminish. Now, the other thing is the sample size. Then, if you're familiar with statistics, I don't need to go into details, but I would just say that these available studies are about 5,000, 10,000, or maximum 15,000 individuals. So, when we are measuring something or investigating something with a small effect size, we have to keep it in mind that we might not find genome-wide significance, or if we suspect gene-environment interaction, which is quite the case with social science phenotypes, then sample sizes collect weight. Now, I painted quite a dark picture about the results of the GWAS, what we can do, but it's not that bad, because the results, what I would like to stress here is that chasing and expecting individual genes coming up very significantly is very unlikely. But we can take the GWAS results further. The most important thing is whatever we got, even if it's very plausible, try to replicate in an independent sample with similar phenotype, and if possible, the genotype in platform being similar as well, but that's not crucial. Or the other tool is a genomic complex trait analysis, when we can take all the GWAS results and to see what proportion of the phenotypic variance is explained by them. Or if we are very cunning, then just use all SNPs which are like we think do some function, or I set up a p-value threshold. It's really just your imagination what you want to do. The next big thing is the polygenic risk score. And this is for an individual, is take the sum of the trait associated alleles and weight by their effect sizes, which are calculated from the GWAS studies. And the next sentence is application is to test for shared genetic etiology between traits is very important. So there was a study when they used schizophrenia polygenetic risk scores, and they found that it predicts 1.5% of the variance in major depression, which doesn't seem to be much, but what I said about the major depression GWAS before, it's quite impressive. Or we can take the replication onto a different level, and we try to see if it overlaps with metabolic profiles, or take it into network analysis, where we expect the enrichment of the genes in the same network.
Thank you very much, Kris.